Okay, in this screencast, I hope to further define some of the definitions involved in cell transport. So cell transport is how various critical molecules and ions get into the cell and out of the cell for that matter. So we're going to talk about some of the definitions, but we're going to give them a little more context and allow you to practice some of the terminology here. So let's start with level 1, concentration gradient. So when you're thinking about a concentration gradient, really what you want to do is think about the amount of, say, an ion or a molecule on one side, and that can be represented visually pretty simply. If we draw a bunch of circles on this side of this artificial membrane, so it's a selectively permeable membrane that will allow these circles to pass from one side to the other. So a lot of cell transport functions on this concentration gradient. So it's pretty easy to see that on this side of that selectively permeable membrane, you have a lot of circles. Those circles can represent many different things that the cell needs, but for now they're just circles. On this side, the concentration or the amount is much less. So here you have a high concentration, bracket means concentration, and here you have a low concentration. So level 1 understanding of cell transport would be to understand that the cell will allow things to move from high to low utilizing this concentration gradient. And because of that, we get to a couple of definitions which I think you should understand. So some of the key terms here. One is diffusion. Okay, so diffusion is the movement, in this case, of the circles from high concentration to low concentration. This is a form of passive transport because it just happens. They go from high to low without the use of energy. So no energy required to do that. We'll cover down, further down in this screencast what osmosis is, but you could imagine if each of these little circles represented a water molecule, which we've depicted like this before. I'm not going to draw all of these, but if this were in fact water molecules, they would undergo osmosis from the left to right side, just as the generic circle would go from high to low. Okay, so cover osmosis down the road a little better, but for now, osmosis goes from high to low as well. Wherever there's more water, it'll move down to where there's less water. Okay, now there are two different types of diffusion. So we're going to add this little wrinkle. Simple diffusion. If this were a biological membrane, in other words, if this was a lipid bilayer, this molecule, this circle, would be able to go right through that membrane. Okay. Facilitated diffusion. Facilitated, there we go. Would be an instance where in that biological membrane you need a protein channel for it to pass through, as in water with aquaporins. Okay, so we're trying to move beyond these simple definitions of diffusion and osmosis, but understand that they're both passive forms of transport which utilize the concentration gradient and go from high to low. One last quick definition and explanation of it, if we go back to this generic circle idea with this side being low concentration and this side being high concentration, is that there's also active transport, which is sort of the opposite of active transport. So with active transport, you're going to go from low to high. Okay, so from this low concentration to the high concentration against the concentration gradient, going low from low concentration, high concentration against that concentration gradient. And the thing with that is that this requires energy to do that. Okay, so going low to high with active transport requires energy. So in order to understand cell transport on a complex level, you have to understand this concentration gradient. Concentration gradient is sort of a level 1 concept. Level 2 would be understanding both the water component and the solute component. So if you remember back to basic biochemistry, a solution, especially an aqueous solution, has two components, solute and solvent. Okay, so the solute, if you were looking at a cup of salt water, the solute would be the thing that is being dissolved in the solvent. So in this case, our solute is salt. Okay, and in the case of aqueous solution, a water-based solution, the solvent would be water. It's always good to stick with water as an example for biological examples because, you know, cells are made of 70% water and it makes a good solvent. Okay, so let's take a look at salt water as a generic example. If you had a beaker or a cup of some kind and you made an artificial cell like we did in lab, let's say the cell is about this big, and I'm kind of assuming that the space inside this cell and outside here in the solution surrounding the cell is about the same volume. So if we filled that beaker up with water and we took a look inside the cell, we could define the amount of solute in the solution that makes up the inside of the cell. We can define the amount of solute in the solution surrounding the cell. So this is getting to the vocabulary that we talked about, tonicity. Okay, and we talked about various kinds of tonicity. Hypertonicity, or being hypertonic, being hypotonic, and being isotonic. So again, 3 definitions that I want to move beyond in this example here and talk about both the solute component and the solvent component and how both move in these examples that we're looking at, in these artificial cell examples. Okay. So let's take a look at a hypertonic cell. Well, let's take a look at an example of a hypertonic situation. Because remember, what this defines actually is the solution surrounding the cell. So we're going to say that the solution surrounding the cell is hypertonic. You know from class that hypertonic solutions have more solute. So hyper meaning more. So if you want to represent a solution that is hypertonic using generic circles or generic pluses as an example, you really have to load up the pluses to show this hypertonic state. So I'm going to go overboard here and show this. Again, these pluses representing generic solute. Everything else surrounding this solute is water. So that's your solvent in this particular solution. And oftentimes I do not draw water molecules. It is sort of, I guess, assumed that this solute is in water. But what the heck, we can draw some water here just to show you what we mean. Okay, so keep in mind that wherever there is more solute, there is less free water. So I'm only going to draw the water molecules that are freely available to move. Okay, so because there's a lot of solute out here, there's not a lot of free water to move. And we went over this example in class. We acted it out. We did a little simulation. But let's just go ahead and depict that like this. Now, because inside the cell there's more solute, or less solute, excuse me, you're going to have more water freely available to move. For those of you listening in that maybe are not in our class, you can take a larger look at a water molecule here real quickly. This is an oxygen atom, and these are hydrogen atoms. Of course, in the famous formula H2O. And water as a molecule is polar, this side being slightly negative and this side being slightly positive. So because of that, it binds really well to ionic compounds, breaking them down into ions. In our class, we went over salt, sodium chloride. So hopefully from this description here, you can see that the solution surrounding the cell has a lot more solvent than inside, right? So I count only 4 here if we're representing this, and I did not count the number of pluses here, but let's just say it's 50. Okay, so because of that, the solution surrounding the cell is hypertonic, making the inside hypotonic, okay, containing less solute. The flip side of that, you can see here, based on all these water molecules inside the cell, is that there's more water inside the cell freely available to undergo osmosis than outside the cell. So what happens here? Well, let's take a look at what happens with the solute and what happens with water in this particular example. Okay, so solute, these red generic pluses, are going to want to move in. So the solute's going to want to move in. Again, it's going to go in from high to low. That is diffusion. Okay? Uses the concentration gradient. So high to low, based on concentration gradient. The flip side to that is that there's more free water inside the cell that will want to come out. Okay, so the opposite will happen with this water molecule. It will also go from high to low via osmosis. Okay, but it's going to want to go out. So it's going to want to go in the opposite direction of the solute. So what will happen to the cell? Well, if you take a look at that, we talked about in class that solute contributes very little to either the swelling or the shrinking of the cell. So now, based on water wanting to leave, so water out would lead to cell shrinking. Okay, so that water moving out will lead to cell shrinking. It's going from where it's highly concentrated inside the cell to where it's lowly concentrated outside the cell, so you're going to get cell shrinking. So in a hypertonic solution, you're going to get shrinking because water goes out of the cell. So really you have to understand the various tonicities of tonicity solutions, that is hypertonic, hypotonic, isotonic, in order to understand cell transport. This is sort of a level 2 concept, and you should be able to, for every example that we went over to in class, put a cell into a hypotonic solution, put a cell into an isotonic solution, and tell me where the solute is going and where the solvent, in this case water, is going. The real sort of cheat way to do it, I mean, it's not a cheat way, it gives you the answer most of the time, is to say that they go in opposite directions. But to really understand what a solution is and how it works, you should understand that there's more free water, thus creating a water concentration gradient where there's less solute. Okay, so it will move from high to low away from where there's less solute to where there's more solute. Okay. Okay, now this level 2 is going to present itself differently at different levels of understanding. So, you know, for starters, I'm fine with depicting situations like this wherein you have that internalized artificial cell and you make a representation of solute with a certain shape. I don't mind that. That's sort of good for beginners. That's the way to do it. So if we were looking at this example here, again, this cell would be in a hypertonic solution. One step up would be to create maybe a percentage. So to understand, to quantify it a little more, a B example of a hypertonic solution surrounding a cell would be to say that this cell is 50%, say, sucrose. To be surrounded by a hypertonic solution, the inside of the cell has to be less than 50. Okay, so let's say that it's 20% sucrose. Okay, so percentage is a way, a more advanced way of understanding, you know, amount of solute in a solution. And then once you have an understanding of chemistry at the AP biology level, you might say, you know, to represent a hypertonic solution surrounding a cell, since you'll understand molarity, you might say that this is 10 molar salt. And the inside would be 1 molar salt, which is a lot, but you get the point. So the inside would have to be less, containing less solute. And you know, these concentrations are just the amount of solute, the amount of dissolved substance per unit volume. So all three of these are fine ways of describing a hypertonic solution surrounding a cell. This is very much for the beginners. This is what we do mostly in regular biology, and then this would be more of an AP biology question. Okay, so level 3 is putting it all together and understanding the fact that in a cell, there's many different things that are moving into and out of the cell at a given time. And one of the best examples to set this up, you'll see this all the time, are these U-shaped tubes with, again, an artificial selectively permeable membrane in the middle. And then we're going to set it up for different rules. So we're going to say, okay, there's a certain amount of water in here, and we're going to put these large circle solutes here, and we'll make this side, the A side, higher in those, and the B side less in those. And then we're going to say, okay, well, since the cell is transporting many different things, let's put, I don't know, some pluses over here. And we'll put this side having more of those pluses than this side. But there are equal amounts of water, okay? And it's sort of like a little game. We're going to say that pluses can cross membrane. Okay, but we're going to say that the circles cannot cross membrane. And then it becomes a little game. So these red circles are going to want to cross, but they can't according to the rules of the game. So from either side, they cannot cross. However, the blue crosses can in fact cross. So what will happen over time, and you break it down step by step, One, let's cover the solute that can diffuse from high to low. And so that will be the pluses. So these pluses can go over here. They can diffuse to this side. And what happens is over time, they will come to a dynamic equilibrium. That is to say, they will be isotonic. Okay, they will equal out. Over time, you will have equal amounts of pluses on both sides. The red circles, however, cannot reach a dynamic equilibrium. They will always be higher on the A side, on the left side. So what happens then? Well, then you have to consider the side that has the higher amount of free water. And depending on how many of these pluses and circles you have, that's going to determine if you can, in fact, have osmosis occurring in one direction. Let's just say that there's more free water over here. That is water that's not bound to the plus or circle solute. What will happen is, since the red circles can't cross to the right, water will move from this side to this side, causing water levels to rise on this side and causing water levels to decrease on this side. So really, this is I know we're talking about cells here, and it happens in cells just the same as it would in this U-shaped tube. You have to really break the problems down and solve it solute by solute according to the rules. Okay, so I hope that helps with your understanding of diffusion and osmosis and gives you a better picture of what those definitions mean.